Hello, my name is Tillotson. I'm from GST Micro, and the title of my talk is Bacteroidota, the keystone phylum of the human gut microbiome. My disclosures are I'm a consultant to Ferring Pharmaceuticals, Spiro Therapeutics, and Taro Pharmaceuticals. I'm an advisor to Parasec Pharmaceuticals. So let's get started. A keystone species is one whose effect is large and disproportionately large relative to its abundance. We all understand that microbial communities maintain a healthy ecosystem, and these systems are ubiquitous in nature and include both plant and animal kingdoms. The role of these ecosystems is vital in human health, and an estimated 500 to 1,000 microbial species, which is around 10 to the 13 cells, are present in our bodies. A little bit of ecology background for you. The species found in any ecosystem are of a variable importance, and as shown before, the term keystone taxa was discussed by Power and colleagues, particularly Dr. Payne, in 1996. They hypothesized that certain taxa co-occur in microbial networks and are highly important in the integrity and structure of these communities. But when a shift or decrease or removal of these taxa leads to a condition termed dysbiosis. More specifically, the GI tract contains the largest number of bacteria in the human. Bacteroidota, are a key player in the healthy gut microbiome. Now, let's talk about the majority of the gut bacteria. We have Firmicutes and we have Bacteroidetes. The Firmicutes, these are gram-positive bacteria with rigid or semi-rigid cell walls, and they're predominantly from the various genera Bacillus, Clostridia, Enterococcus, Lactobacillus, and Ruminococcus, and many of these can form spores. In the Bacteroidetes, this includes approximately 7,000 different species. They're all gram-negative, and they range from strictly anaerobic Bacteroides to strictly aerobic, such as Flavobacteria. Other species in this family are the Allistopes, Parabacteroides, and Prevotella. I'm not sure you can see this particularly well, but this is a quick guide to Bacteroidetes taxonomy. We have a domain on the far left, phylum, class, order, family, and genera. And the genera, Bacteroides, is the upper one of those green bands on the right, and you can see within them Bacteroides fragilis, Bactertheta omicron, vulgatus, and so forth. And they will come up as I go through the presentation. And just to give you a feel for the Bacteroides species, this is a list. There are 40-something species on here, and there are some what we call sensu stricto. These are the key ones. These are the main proven Bacteroides, Cacac, Fragilis, Uniformis, and others like Ergerthii and Theta omicron. But as you can see, the Bacteroides is a fairly complex group of organisms. Now, what do Bacteroidetes do in the human gut? They have a variety of activities, and what I'm going to do is go through these different activities to give you a feel for the impact of these organisms, and perhaps the reverse of that is what happens when these organisms are not there. So let's move forward. You can see the pretty diagram there on the right, and you can see that there are various bullets, highlights, and I'm going to go through each of these in some detail. But on the bottom left, you can see a reference in Open Forum in Infectious Diseases that I published late last year, which goes through each of these particular topics. So let's start at the top of the diagram, sphingolipids. As many of you know, sphingolipids are membrane components, and they're important in signaling molecules that regulate inflammation and immunity. And a variety of Bacteroidetes-derived sphingolipids includes ceramide phosphoinositol and deoxysphingolipids. And these molecules are critical for maintaining homeostasis and symbiosis in the gut. Number 2, Bacteroidetes and the gut-brain axis. I think we've heard a lot over the years about the relationship of a healthy gut or an unhealthy gut and how it can affect brain function. And here, gut microbes, including the Bacteroideae, produce metabolites, which have highly neuroactive potential, such as neurotransmitters, including gamma-aminobutyric acid. And this is the main molecule that's involved in the neurotransmission within the brain. Multiple Bacteroideae species produce GABA through a highly conserved glutamate decarboxylase system, which involves glutamine and glutamine GABA antiporter and a potassium channel. All of these interact, produce a flow of GABA to the brain. And all 4 genes of this GAD system are present in 90% of the Bacteroidea's genomes. So you can see the inherent ubiquitous importance of the Bacteroideas in the gut-brain axis. Within this group, Bacteroides ovata synthesizes GABA and modulates the levels of tryptophan and glutamine within the neural system, important cofactors. Number 3, colonization resistance. There is a system called the type 6 secretion system, which is a molecular conduit that enables gram-negative bacteria to translocate effector proteins into neighboring cells. And this is important in that it enables colonization resistance and commensal stability. And by that I mean, if you don't have the Bacteroides and they are not able to produce these particular systems, the type 6, then colonization with problem organisms like Clostridium difficile can occur. And the stability of the commensals is also important in terms of maintaining defense mechanisms. Bacteriocins. Bacteriocins are a fairly complex set of molecules and produced by Bacteroides. It's a family of broad spectrum peptide toxins produced by these species, and they have an inhibitory effect against diverse Bacteroides and Parabacteroides, including those members of the environment and pathogens. So Bacteroides have certain housekeeping mechanisms, and these peptides control which organisms are present and not. Number 5, the glucuronidase enzymes are critical in terms of digestion of complex molecules. Bacteroides uniformis encodes a structurally and functionally distinct glucuronidase, which has specific roles in processing drug glucuronide and glycan substrates. It highlights their broad structural and functional diversity amongst these enzymes within a single human gut microbe. So Bacteroides uniformis is particularly important in terms of managing and controlling the digestion of key substrates. Within infections like Clostridium difficile, Shigella and Salmonella, we see significant intestinal injury, whether it be direct lipopolysaccharides such as Salmonella, or toxin-derived injury such as C. difficile and Shigella. Bacteroides vulgatus, FTJ-S7K1, was found to protect against lipopolysaccharide-induced acute intestinal injury. And this occurs by modulating cytokine production in the colon tissue and regulation of the structure of the gut microbiota. So B. vulgatus is a significantly beneficial member of the microbiome in the gut. This is a particularly important feature because I don't think until recently we really appreciated the role of the interaction of some organisms and their preventative capabilities. And here we're looking at Bacteroides theta iota omicron, which has been found to inhibit the colonization of C. difficile and inhibit inflammation of the colon, actually by attenuating lymphocyte infiltration in the mucus layer, and therefore leads to decreased edema in the submucosa. So here we have a Bacteroides species playing housekeeper to the adhesion of C. difficile in the gut, in addition to modulating the immune system. Now, this is a particularly interesting one because, as I've just mentioned, C. difficile causes infection by virtue of two toxins, primarily toxin A and toxin B. Here, It's been demonstrated that the polysaccharide capsule of Bacteroidota omicron plays an important role of colonization resistance by suppressing the toxin release from C. difficile. So in other words, it stops the problem at its origin. Also, this lipopolysaccharide maintains the production of beta-lactamases, which can help maintain the balanced microbiota. By virtue of the beta-lactamases can destroy antibiotics that come in. 9. Elevation of metabolic dysfunction. The relationship of the microbiome into a variety of metabolic diseases has been a point of considerable interest over the last few years, particularly conditions such as obesity, diabetes, and so forth. And Bacteroides dystosonis has actually been shown to alleviate obesity and some other metabolic dysfunctions via the production of succinate and secondary bile acids such as lithocholic acid and ursodeoxycholic acid in the gut. So it may be possible at some stage to use dystosonis, Bacteroides dystosonis, in some form of management of obesity and other metabolic conditions. Now, the modulation of the immune system. I've already mentioned this in passing, but Bacteroides fragilis, polysaccharide A, clearly has a role on the maturation of the host immune system. Equally, Beta-iota omicron modulates the immune responses to a particular dominant antigen in the intestine. And Bacteroides vulgatus, lipopolysaccharide, can activate adaptive immune responses and TLRs, which clearly are important in early recognition of invading or dysbiotic organisms. Now, what I've mentioned so far are the benefits, the plus side of the Bacteroides, the Bacteroidota. But as many of you will be aware, the Bacteroidetes are villains as well. How do they become villains? Well, they all produce what's called a capsule, and that capsule can be composed of a variety of molecules, including various glucose molecules, carbohydrates, and various lipopolysaccharides. By virtue of this, they're able to evade the host immune response. Also, Bacteroides fragilis has been shown to produce enterotoxins, and these enterotoxins exert an effect in the bowel and lead to significant edema and outpouring of inflammatory response. TF, Bacteroides fragilis toxin factor, is a recognized problem and clearly one that is involved in some of the infections that I'll mention in a moment. Here we come to LPS, endotoxin or lipopolysaccharide. And as I'm sure many of you realize, that LPS is in of itself a significant stimulant to the immune system and can lead in excessive amounts to sepsis, septic shock. So we've got 4 features here. And finally, aerotolerance of Bacteroides. I think we all regard Bacteroides, especially B. frag, as being an anaerobe. There are some Bacteroides species that can actually tolerate a certain amount of oxygen and therefore thrive in environments that we would not expect. And those, in combination with other organisms such as Escherichia coli, Klebsiella, can lead to complex multi-pathogen system infections. So, Bacteroidetes infections in adults, there are plenty. I think one of the more common ones are perforated and gangrenous appendicitis. Gynecological infections have been reported. Skin, soft tissue infections, frequently in the diabetic patient that has a certain amount of fecal contamination, but equally we can see these types of infections in normal immune competent individuals as well. Rarely, but has been observed, endocarditis and pericarditis. This is, as I say, a fairly rare occurrence and one that many microbiologists would not be looking for. But nevertheless, reports have occurred. Bacteremia. I think many of us appreciate that, organisms that can emerge from the GI tract can get into the bloodstream and lead to significant problems. Those organisms that are in circulation can also land in the joints. And as there have been several reports of septic arthritis, usually as a sequelae to bacteremia. Rarely, but still observed, brain abscesses and meningitis. Inflammatory bowel diseases. There have been a relationship observed with ulcerative colitis and Bacteroides infections, and clearly it's an interaction that we don't fully understand, but I think that some of the Bacteroides species, by virtue of producing different toxins, can actually elicit an immune response that leads to a flare in IBD. I haven't listed there, but I think most of us appreciate that gastrointestinal abscesses are one of the major problems that we see with Bacteroides, but they are invariably in mixed infections, as I say, with the Enterobacteriaceae, Enterococci, and so forth. So Bacteroides can be a bad guy, and I'm sure many of you realise that. So I'd like to summarise, really, just going through some of the pluses and the minuses. In the beneficial column, plasticity, a topic I've not mentioned, but it refers there to the ability to transfer various genes between organisms, and that can carry things like antibiotic resistance and some other virulence mechanisms. So plasticity shows the ability to send and receive various genes. We've seen the broad metabolic capabilities. Again, I've not gone into depth and detail, but short chain fatty acid production is essential in some of the regular pathways, such as gut-brain axis. We look at changes in the bile acid profiles from primary to secondary bile acids. Again, this is a lecture in of itself, and I think it's important that we appreciate that the role of a healthy gut provides a lot of beneficial metabolites, and I think those are demonstrated in terms of maintenance of gut-brain function. The effect in infection is significant. We have been through the virulence mechanisms, and I think any one of those I'm sure you become aware of. In terms of colonization resistance, I think one of the major things for me is the ability to inhibit Clostridium difficile, either colonization, more importantly, C. diff toxin suppression, which I think is a novel concept that many of us didn't think of. Immune modulation is important. I think I've described that the prevention of immune function in the bowel can lead to significant edema. Reduction of inflammation has been mentioned already by organisms like B. uniformis, and there is also discussion, fairly early stage yet, but Bacteroidetes may well have a role in anti-cancer immunity. On the downside, Bacteroidetes produce an enzyme called neuraminidase, which leads to priming the site for infection. We've spoken about type 6 secretions, which antagonizes against beneficial Bacteroides by the production of type 6 from B. frag. Antimicrobial resistance. It occurs through horizontal gene transfer, as I mentioned, and there is a myriad of antibiotic classes to which Bacteroides have become resistant to. Oncogenesis, I think I've already mentioned that there's a lot of interest in work in the field of the role of Bacteroides in terms of cancer, both in terms of eliciting cancer, but also in terms of inhibiting some of those particular cell lines. And then enterotoxin production. Some strains of Bacteroides fragilis produce enterotoxins, which are not dissimilar to the ETEX that I'm sure you're familiar with. I've put a couple of references here that may be of use for you to go back and have a look at. One is in Open Forum Infectious Diseases, and the second one is in anaerobe. And anaerobe is where the title of this talk came from, and without wishing to be big-headed, those are two papers that I was involved in. So thank you for your time and attention.